When we talk about cells and cell function, we really have to talk about the cell membrane, because the cell membrane is sort of what sets the stage for what the rest of the cell is able to do or to not do. And what we see here is a drawing of a cell membrane, and we have two different layers to this system. So we have an outer layer here and an inner layer of these little fats called phospholipids. And what we have is we have heads and we have tails on those phospholipids. And so the little heads to the outside, the tails to the inside. And this actually means that the heads are hydrophilic. That means they interact with water. They tolerate water. And the tails are hydrophobic, meaning they repel water. So the cell membrane is actually able to get a waterproofing kind of concept going on in terms of the tails to the middle of the cell membrane, but also the ability to interact with their aquatic environment on the outside and on the inside with the water interacting heads. We also notice here a number of orange structures scattered throughout, and these are proteins. And these proteins have a number of different jobs. One of those you can see here in the middle is an integral protein, and that's going to be involved in basically as a channel or a gate. That if it opens, it allows certain things to go through the membrane. If it closes, it prevents those things from going through. Some of these proteins are used for attachment to other cells or to other areas of the cell. The proteins we see with the yellow chain, the carbohydrate chain structures there, they're labeled in some of them as glycoproteins. Those are, or glycolipids on some of them as well, those are name tags for the cell. So when the immune system comes along, it reads those name tags, and that name tag then identifies that cell as being a liver cell, or a skin cell, or a bone cell, or something like that. And then the immune system says, okay, no problem, because I recognize your name tag. And when we get to the immune system in a later chapter, we'll talk more about that. But cellular identification is essential to your immune system's ability to protect yourself. It learns to recognize what is you and to leave it alone. And if it doesn't recognize you in the form of a bacteria or a virus or some other problem, then it is able to destroy selectively those cells that it does not recognize, or those particles that it does not recognize. Let's talk about how things move back and forth across that membrane. And this is really how the cell sets the stage for everything it can and cannot do is based on what the cell membrane allows or doesn't. So in the left image here, we have our starting conditions. We have our cell membrane down the middle in blue, and inside the cell we have some particles of things. Let's just call those sugar for the moment. It's probably not, but we'll just call it that. Is our purple, and then our red and gray sphere structures, that's water. So we have a certain amount of these particles on the inside of the cell, and a certain amount of those particles on the outside of the cell. What we find is the concentration of both water and this purple molecule are greater outside the cell than they are inside. And this is not at all what the cell wants. Typically, biological systems are looking for equilibrium. They're looking for balance, and what they want to see is the same amount of something on one side or in one area as you would find on the other side or in the other areas, whether within a contained environment or in this case where we're looking at what's on one side of the wall compared to what's on the other side of the wall. So in this case we have less inside the cell, more outside. And what we're going to see happen here is called diffusion. And after a certain amount of time, what we see is that we have more purple has moved in and more water molecules have moved in. At the same time, some are moving out. So they're constantly moving in and out, but typically they're going to move towards equilibrium in the direction that they need to achieve equal concentrations on both sides. That's really what we're aiming for here. And so movement of particles from high concentration to low concentration in an effort to achieve equilibrium is diffusion. Now, there are a couple of kinds of diffusion. The first example here is just, for the most part, plain diffusion with the purple molecules moving back and forth. And if they are big enough, or rather if the purple molecules are small enough, so the openings in the cell membrane are large enough, then those purple molecules, as we see here, move freely back and forth. Water is also moving back and forth here. And for non-water particles, diffusion is going from high to low. Again, whether it's in the same container or whether it's crossing some sort of barrier. When we talk about water moving from areas of high water concentration towards areas of lower water concentration, that's called osmosis. So osmosis is diffusion of water across some sort of membrane. Here we see a couple of different examples of cells attempting to achieve equilibrium with their environment. And achieving equilibrium with your environment is not always a good thing. That's not always going to produce desired outcomes. Our first set of images here in A, the cells in the beaker and the cells in the actual photograph, there are red blood cells. And that is an isotonic solution. What that means is that the concentration of, we'll generically say salt, but all dissolved particles, we'll say, the concentration of those salts and other things inside the cell is about the same as the concentration outside the cell. So we don't really have large amounts of stuff moving back and forth. We've roughly achieved equilibrium, and that's what we want. And the shapes and sizes of the cells there, note that all of these are pretty much at the same level of magnification. That's what you would expect to see. In option B here, a hypotonic solution, what we have is the cell is saltier, or it has more dissolved particles in it than does its outside environment. And in this particular case, if the particles are too large to move readily into or out of the cell, simple diffusion won't do the trick. We can't just move some particles, in this case, out of the cell to balance it out. So if those can't move, water typically has to move to balance things out for us. So in this case, the cell was saltier than the surrounding fluids, meaning the water went from the fluids into the cell, trying to balance out the higher concentration of salts inside. What we get here really is the cell swells. So we see the exact same level of magnification, but these cells are a good 50% or a little better bigger than we saw before because they're becoming bloated with water. And when that happens, you're going to have some serious problems. Those red blood cells will not freely circulate through the body because they'll get stuck in capillaries. And we'll learn about capillaries later, but they're typically so small in diameter that red blood cells kind of squeeze through one at a time. And if you've dramatically enlarged your cell size, that's just not going to work. Scenario C here is a hypertonic solution. So in this case, the external environment is more salty than the cell. In this case, the particles cannot move into the cell to balance things out. So water has to move out of the cell, trying to balance things out in the external environment. And what you see there is the cells shrivel up and collapse. This is not a good thing from circulation standpoint either, because they're much more likely to clump up and stick to each other in this rather rough and lumpy condition, meaning you have more likelihood of blood clot formation to occur. And if you've ever experienced a dehydration headache, whether it was because you just didn't drink enough for several hours to a day at a time, or perhaps a hangover, is an extreme dehydration headache, in that case caused by alcohol causing the hypertonic solution to occur. And what you have is your red blood cells shrivel up, they clump up, they don't do a good job of carrying blood to the system, and your brain becomes deprived of oxygen. And so really a dehydration headache is your brain doesn't have good blood supply, and your solution is always to, no matter what the cause was, to rehydrate the system. So in this case, consumption of more liquids would reduce the saltiness of the fluid in the body already, which would then allow water to move back into cells that had moved out of the cells trying to achieve equilibrium in that system. So the hypotonic solution in the middle would occur if you're overhydrated. The hypertonic solution would occur if you are underhydrated or dehydrated. If on, let's say, a hot summer day, you choose to drink a quart of fluid, which is a pretty good size amount of fluid to drink all at once, and then you are going to do that within, let's say, a short period of time, let's say a 10-minute window. That's chugging it down pretty good, but what you're going to find is if you chug in plain water, you're going to experience a hypotonic scenario. The water you're dumping into the stomach would be much less salty than the stomach cells, and because salt typically can't cross cell membranes, we look at water moving into the stomach cells very rapidly, blowing them up. You then feel nausea and the need to vomit, and you feel very unsettled with that because your stomach is literally starting to drown and be overwhelmed with these fluids, and it's screaming at you to stop dumping those fluids in there. If you were to consume that same amount of fluid as, let's say, Gatorade, Powerade, or something like that, you wouldn't necessarily experience that, all other things being equal, because those solutions are what we consider osmotically balanced. They're roughly the same salt concentration as your cells normally are anyway, so you don't trigger that massive a movement. Really, we're talking about them being an isotonic solution. So the Gatorade would be roughly the same as the cells, so that's an isotonic scenario, and that's back where we wanted this whole thing to be to start with. If you would swim in a saltwater body of water, let's say, you would experience to some degree a hypertonic solution. So there you would see the shriveled cells, and that would be your entire body becoming experiencing hypertonic situations and losing water to that environment. If you spend a lot of time in a freshwater pool, you would see your fingers swell. Now at first they would look wrinkled, and they are wrinkled, but not from loss of water like you would see in the ocean, but they're wrinkled because they're swelling unevenly. And once you give it some time to equalize, you just have fat fingers. And that would be your body experiencing this hypotonic solution. The body's saltier than the freshwater pool. It blows up with water, and away you go. Typically a couple of hours of freshwater exposure, and you feel the need to go to the bathroom and urinate because you've got all this water that's flooded into the system, and the kidneys have determined that's too much, and they're dumping it out. So diffusion in any form, whether it's standard diffusion or whether it's diffusion of water in the form of osmosis, are free processes. They don't take any energy because the concentration itself drives the process. So no energy expenditure occurs. What we have here is called facilitated transport or facilitated diffusion. And that is the same idea of going from high concentration, in this case outside the cell, these little sugar molecules. So we have a concentration of 4 outside and 2 inside. That's a differential. But in this case, the sugar molecule is too large to simply squeeze through some small openings, and it's not made out of fat. We should distinguish at this point that fat-based particles very easily diffuse across the membrane. So they dissolve their way right through those phospholipid layers with no resistance. But glucose is not fat-based, so it doesn't do that. Water-based things, if they can't squeeze through, must be passed through in a protein. So this particular protein here is moving glucose in. And so it's not engaging in energy, it's simply like opening a door and allowing that diffusion to open through the door. And as long as the door is open, diffusion occurs. If the door is closed, obviously the diffusion would stop. So this is the way that your body regulates blood sugar levels, is it either allows sugar to go into cells or it allows sugar to go out of cells into the blood based on your needs. And as long as you don't have diabetes, typically your body does a very good job at this facilitated diffusion process, keeping the sugar concentrations level where we would like them to be. But again, this was free. We didn't use energy for it. We simply opened a door and let it happen. We closed the door and stopped it. In this particular example, this is called active transport. And active transport involves pumping things back and forth using energy. So here we see ATP on the inside of the cell. That's our energy molecule. And it is releasing some of its stored energy and using that stored energy to open the door. So this is not a freely swinging door. This is a very tightly closed door unless you use energy to unlock it. And when you do that, then we can have sodium and potassium come in and out of the cell by pumping it back and forth. So that actually takes energy. That is somewhat costly to carry that out. We would do this if we wanted the concentration to go against the concentration gradient. So what we see here is we have a concentration of 3 potassium outside, 4 potassium inside. And normal diffusion would say we want the potassium to go from inside to outside. But in this case, we don't want that. We want the opposite to occur. We want to build a greater concentration on one side, cause a lesser concentration to occur on the other side, and that's why it takes energy, because that is not free. The same thing is going on with sodium. We have more sodium inside to start with than we have, or rather we have more sodium outside to start with than we have inside. So normally sodium would want to come in, but we're actively pumping it out here. This is the sort of thing that we would see happening all over in the body, with muscle contraction, with nerve signaling. This sort of swapping of things back and forth is going to occur. And also in the kidneys, we're going to see a lot of moving things back and forth in opposition to the concentration based on what the body needs to achieve. So active transport requires energy to carry it out. Here we have a really cool idea going on, and this is called endocytosis. Endocytosis means to take something inside, so the endo means to take in. Cytosis means to take it in across the cell membrane, having something to do with the cytoplasm inside, the gel matrix inside. So in this case, a particular form of endocytosis, this one would be phagocytosis, and this is eating something by engulfing it in cell membrane. So you can see what's happened here is the plasma membrane has formed a little pocket, sort of sucking in that particle, and then it wrapped that pocket completely around and pinched it off inside. So now it's floating inside the cell in its own little membrane package. So it just ate it. If we were talking about a white blood cell, this is how they eat bacteria and other foreign particles. They swallowed inside basically by giving it a membrane hug and pinching off that membrane sac inside. Once it's inside the cell, it's contained, and you can do whatever with it that you want. So this one might actually break that down for energy. If it was a white blood cell eating a bacteria, it would then use some lysosomes and some other things like that to kill that particular bacteria and cause it to no longer be a concern. But cell swallowing of particles is phagocytosis. Another form of endocytosis is called Pinocytosis, and this is consumption of liquids, perhaps with dissolved something in there that you're looking for, but liquid consumption by a cell in these membrane-bound packages is Pinocytosis. And then a third type of endocytosis here, and that's receptor mediated. So that means that this particular occurrence, this swallowing, wouldn't occur unless there was a particular trigger there to activate the receptor proteins to form that little package. So if there's no trigger, then this receptor mediated process doesn't happen. So all three of those are really just different ways the cells can take something from outside the environment of the cell and take it inside the cell, either to use it or to kill it or to do something else useful with it. So over here on the left, we have that process occurring, and this looks like some kind of phagocytosis. And if we go down here and follow it, this little vesicle, this little package we formed from the membrane, comes down here. In this case, we're adding in some lysosome. So that indicates this is some sort of foreign particle or something that needs to be broken down. And so that digestive enzyme gets dumped in there on it, and we can see here that enzyme breaks it down into little pieces. So whether it was destroying a foreign particle or breaking it down for consumption or some other function, it involved, in this case, enzymes being dumped in there. And so that makes it a sort of a self-contained chemical reactor inside that membrane. It's certainly a safer way to do things. On the other side of this schematic here, we have secretion of something. So if endocytosis would take something into the cell, exocytosis will remove something from the cell with the same basic mechanism, and that is to form a vesicle or this membrane wrapping around your particle, and you can see here, send it out to the outer surface of the cell and then release it into the external environment by having that little package fuse with the outer membrane and open up. And so you can do this with, in this case, it made some proteins. Those proteins needed to go to some other part of the body, and so it was packaged in that little vesicle, sent to the surface, and released with exocytosis. So endocytosis is taking something in by swallowing, exocytosis getting rid of something by spitting out, but both of them using those membrane packages to do that. When we talk about the metabolism of a cell, this gets very complicated very quickly, and the first piece of the puzzle is the use of enzymes. So you've heard of enzymes before, and you know they're important, but you might not really know what they are. Enzymes are proteins whose job is to encourage chemical reactions. And what we see here is that if we didn't have enzyme present, for this particular reaction to occur, follow the blue line, it takes a certain amount of energy to get that done. But if we had enzymes present, we can see that it would take a much lesser amount of energy to get that reaction done. So enzymes reduce the energy of activation, or the amount of energy it takes to do the reaction. They also speed it up to allow it to happen more quickly, because for a reaction to occur, two particles or two things need to come in contact with each other, and then for them to actually react once they contact, they have to be lined up exactly right. So enzymes ensure that every time something contacts its other matching particle, the alignment is perfect. So it makes it much more likely that every time two things come together, they do react because they're always aligned properly. So reduce the amount of time it takes, reduce the amount of energy it takes, and you get a much more efficient process. If we think about the food you eat, and if we think about probably your favorite category of food would be carbohydrates for many people, think about bread. You take a nice bite of bread. How long is it in your mouth being chewed before it seems like it's almost all gone? And the answer is it's a matter of seconds. So in your saliva, there are a number of very powerful enzymes whose focus is digesting carbohydrates. So with those enzymes, you can digest many carbohydrates in your diet in a matter of seconds. If you didn't have those enzymes present, digesting that thing that took a couple of seconds otherwise might take hours to days to occur. So if we look at a meal is digested start to finish in roughly 24 hours. We've consumed it, we've processed it, got everything out of it we can, and it's ready to be disposed of, whatever happens to be left over. So with enzymes, we're looking at that 24-hour period. Without enzymes, we would be looking at weeks to digest a meal. And obviously that wouldn't work because we don't have time to wait that long to get the energy. We don't have space to have that much food in the digestion process to keep us supplied with energy. So it just really wouldn't work. So with enzymes, life occurs. Without enzymes, life really doesn't work so well. And let's say you overheat, and that can actually cause a heat-induced heart attack because the heart enzymes would become inactivated. So if conditions in the body get off, whether it's the temperature being too high or too low, salt concentration gets too high or too low, the pH, the acidity gets too high or too low, any of those things, and some other things too, if those get off too badly, then enzymes stop working. And if you've ever experienced heat exhaustion or heat stroke, you would know exactly what we're talking about here. Your enzymes reduced their ability to function because they lost their proper shape, and then you felt really horrible and perhaps took a few weeks to recover from some of those types of events. Most of the cell's metabolism occurs in the mitochondria. And the mitochondria is considered the powerhouse of the cell. And within that mitochondria is where we're going to see most of the rest of this reaction here. So what we start with is glucose. So glucose and oxygen come out of our blood vessel over here, a little capillary, and enters the cell. The glucose goes through that protein door, and the oxygen just wiggles its way through the cell membrane through standard diffusion. But what we have here is we take that glucose, and I'm not going to give you this reaction in great detail because it would take a couple of hours to work through it in great detail. But the general idea is in glycolysis, you take that sugar and you break it down into smaller pieces. From that, you see at the bottom, we got a little bit of ATP, a little bit of energy. And the whole point of this is to convert sugar and the energy that it contains into a format that the body can actually use. Because the body can't directly apply sugar to a problem, but it can apply ATP to any particular thing it wants to do. So your metabolism is taking the energy in the form of sugar, proteins, fats, whatever it happens to be, putting it through this cellular respiration process to convert it into ATP, which is readily usable by all other body functions. So in glycolysis, we converted one molecule of glucose to 2 pyruvates. We got 2 ATP. We then went to the next step occurring inside the mitochondria called the citric acid cycle, in which we generated 2 more ATP. And so we made 4 ATP up to this point. That's great, but that's not very efficient. And then we get down to the third step in this process. The ingredients from the citric acid cycle go to the electron transport chain, where you can see that we get a much greater return on our investment. We get somewhere between 32 to 34 ATP in the electron transport chain processes. Collaboratively, we add all these up, we're looking at somewhere around 36 ATP from every single glucose molecule that goes through this process. So one sugar molecule converts into 36 energy molecules that the body can actually use. Now along the way here, we've got this oxygen, and it's been floating around, but it's not actually used until we get to the electron transport chain. Then the oxygen is actually used in the process and actually then forms carbon dioxide, which we would then breathe out eventually through the lungs. But the oxygen is there to pick up the waste products. So really oxygen is just a superpowered janitor picking up the waste products from these reactions and then helping to dispose of those waste products in a format that the body can handle. So you know about oxygen being so essential to life, and you think it's some big sophisticated thing, when really all it is it's there to clean up the mess at the end. And it turns out that if you don't clean up the mess within 4 minutes to 5 minutes, then you're going to die from lack of oxygen. And that's just you died from a buildup of waste product and then a resulting lack of energy produced. So if you didn't have enough oxygen to clean up that mess, you couldn't make those last 32 to 34 ATP. And you really wouldn't even make the 2 ATP from the citric acid cycle. So if you don't have enough oxygen to fully complete this process and clean up the mess, then you really only get the 2 ATP from glycolysis. And you can do that process without having to complete citric acid cycle and electron transport chain, but you can only do that for a short period of time. Because the waste product, instead of being carbon dioxide, which we easily breathe out, the waste product becomes pyruvic acid, which then can cause muscle cramps and things like that building up in the body, and it can be a real problem. Now that process we just went through there in very, very, very simplified form was for carbohydrates. So glycolysis, citric acid cycle, electron transport chain. Fats and proteins also go through this process, but they differ in where they actually enter the process. Once they're in the process, the steps happen in exactly the same way. But as you can see here, the carbohydrates go right through the middle in that step. If we had the proteins over here, it could go through glycolysis, citric acid cycle, electron transport chain. It could skip glycolysis and go straight to the citric acid. It could skip the intermediate, go straight to citric acid, and then from wherever it enters, go on through. Fat could go in at glycolysis. It could go in at that intermediate stage before the citric acid cycle as well. So again, the difference is where in the cycle they entered. Once they're in the cycle, the steps work exactly the same way. And if you want to appreciate that a little bit more, feel free to read that little section in the book there on the cellular respiration. I'm not really going to expect you to understand it in great detail or to be tested on it heavily, because it's a fairly complicated process, and we would better spend our time on some bigger picture items later on down the road. So just appreciate it is complicated. It is effective as long as you have oxygen and everything works nicely then. And it's differing slightly based on whether it's a protein, carbohydrate, or fat in terms of where it enters the cycle. Hopefully this was informative. You learned a lot and it all made sense. And we'll see you next time.